Vectors in molecular cloning can be broadly split into low and high capacity vectors. Anything with a capacity of less than 50 kb can be considered low capacity. This includes plasmids, phagemids, bacteriophages, and cosmids. High capacity is anything from 50 kb to the megabase range. This includes artificial chromosomes, BACs, phosmids, YACs, PACs, and other derivatives. To make the information digestible, I will split the discussion on vectors into 3 videos. Just to lay out the basics, a vector is any piece of DNA into which foreign DNA can be inserted and propagated, which means you can make copies of that inserted DNA. Vectors were used to copy DNA before PCR was invented. Additionally, the use of vectors was also realized in DNA sequencing, probe construction, cDNA library generation, and manipulation or engineering of DNA. All this could also be used to make proteins. Plasmids are the backbone of genetic engineering, so it is important you know your plasmids. In nature, they're extrachromosomal DNA found in prokaryotes. They're considered non-essential to bacterial survival. Typically, they have genes that encode for antibiotic resistance, toxins, or the ability to use compounds made of sulfur, iron, and many other things. Most plasmids in nature tend to be circular and double-stranded. This circle can be intact, which is called closed covalent circle, or one of the strands can be nicked, so instead only one strand is intact. This is the open circle or nicked form of a plasmid. Quite often, these intact circles inside a bacteria take up an entangled configuration, which we call the supercoiled form. If you extract plasmid from a bacteria and run it on an agarose gel, you expect at least 2 bands that correspond to nicked and supercoiled forms. The supercoiled form runs fast because it takes a compact and densely twisted form. The nicked form is the bulkiest because it cannot supercoil, but it still has one strand intact, so it runs slow. The linear form, if present, has both strands cut, so it runs a bit faster than the nicked form. Sometimes it is also possible to have intermediate varieties of supercoiled DNA. This totally depends on how you prep your plasmid. Most standard lab protocols do not give you visualizable amounts of intermediate forms of these supercoiled plasmids. Sometimes it is possible to see them. Or you just add a bit of topoisomerase to your plasmid prep at the end, and you will see different coils and twists in your plasmids, and they run at different lengths on a gel. They're called topological isomers. Plasmids are not necessarily circular DNA. It is just that applied molecular biology doesn't care about linear plasmids. The linear plasmids typically have their ends protected by hairpins, or they have proteins, just like telomeres, to protect their ends. Since they're complex, we don't use linear plasmids as vectors. So I will restrict the discussion of plasmid vectors to only circular DNA. In a bacteria, plasmid can be maintained at fewer or many copies. The low copy plasmids are called stringent plasmids, and the high counts are called relaxed plasmids. The correlation is usually that larger plasmids tend to be low copy, and the smaller ones are high copy. This makes logical sense, but the size is not a true factor for copy number changes. As we will see, there is something else. The bare minimum skeleton of a plasmid, a natural plasmid, is the origin of replication, because you need to replicate the plasmid. The origin also ensures the type of host bacteria in which the plasmid can live. It may be that plasmids that live in E. coli are not compatible with strep bacteria because of the origin of replication. Apart from the origin, they have at least one gene that confers a phenotype to the bacteria. And I listed some of these phenotypes earlier. This means the majority of the DNA outside of this skeleton is typically trash and can be removed without any problems. Based on the origin of replication, the plasmid can be a restricted plasmid, which means it depends on a compatible host. The Ori-type plasmids are compatible with Ori-C containing bacteria, which is the E. coli. The name of this natural plasmid is COL-E1. In contrast to the restricted type, you have promiscuous plasmids, or broad-range plasmids, which can propagate in most types of bacteria. This usually means that broad-range plasmids are more independent. They don't depend on the host. This means that there are additional genes in a plasmid that make this plasmid independent of the host enzymes. This also means that broad-range plasmids are bigger and will have fewer regions that can be trashed. Our discussion will be centered on the COL-E1 plasmid, and not so much on the broad range. But let's see why size of a plasmid really matters. The problem starts with the energy burden, which comes from the fact that replication machinery has to double time between replicating the plasmid and the bacterial genome. The E. coli genome is about 4 megabases, and it can only fit about 1% extra DNA before it becomes a problem. Usually, exceeding this limit means that the bacterial growth will be slower because DNA replication takes longer since the enzymes are limited. The bigger size of the natural plasmid correlates with extra genes as well, which are responsible for plasmid segregation. Bigger size adds stress on the cells, so there are special genes like partitioning genes that help in plasmid segregation. Bigger plasmids also have a complex origin, demanding the need of extra proteins on the plasmid itself. The broad-range plasmids and their complexity also correlates with copy number. The PUC series plasmids, which we will see later, carry a max of around 10 kb and are present in at least 500 copies. In contrast, some of the bigger plasmids, like R6K, SK, which are around 30 kb, have way fewer copies. Large plasmids are also difficult to insert into a bacteria, and also quite annoying to handle. They're still used, but you pick some special bacteria that can handle these larger plasmids. As I said, most of the molecular biology predominantly relies on COL-E1 derived plasmids. And it is useful to understand how this plasmid is actually physically maintained or replicated in an E. coli. Understanding replication also tells you why a plasmid number has a limit. The COL-E1 plasmid origin is around 500 bases in length. Within these 500 bases is a specific spot called ORI. This is where the first dNTP is added to start the process of plasmid replication. Think of this origin like a TSS, where other factors come together to define its existence. These factors are 2 transcription units, or RNAs, that are part of this 500 base region. At some point, they were also referred to as the L and H RNA, but now the nomenclature is RNA 1 and 2. The RNA 2 is about 550 bases upstream of the origin spot. The RNA 1 is 100 bases downstream of RNA 2. So both of them overlap for about 100 bases. These transcription units have promoters which are recognized by RNA polymerases. So imagine RNA 2 being transcribed first. At the 5' end, RNA 2 has a bunch of inverted repeats, suggesting that these regions can form secondary structures like loops and hairpins. Therefore, as the polymerase moves, the RNA gets folded into specific secondary structure. Shortly after reaching the origin, the RNA polymerase terminates transcription, but interestingly, the RNA2 remains bound to the template DNA. The secondary structures formed at the 5' end of this RNA have a ripple effect on stabilizing the RNA-DNA hybrid at this spot, which will become the origin. To be precise here, the RNA at the extreme 3' end is not in a hybrid state, so you have a bit of a dangling 3' end. This free RNA at the end is recognized by the RNase H, and it gets cleaved, leaving behind a fresh 3' end. This new 3' end becomes the starting point for DNA polymerase recruitment, and eventually a replosome forms, leading to DNA replication cycle. In case you haven't noticed, this style of initiation is very similar to the mitochondrial DNA replication. In plasmids, this successful replication initiation is limited to only a few events, which determines the plasmid copy number. So what exactly imposes this limit? The answer is RNA 1, which is transcribed in the opposing direction of RNA 2. RNA 1 and 2 overlap for a short 100 base region, and this is where all the fun happens. In real time, both RNA 1 and 2 are transcribed without any bias. They will pair at the 100 base overlapping segments. In this hybrid state, the RNA2 cannot form secondary structures at the 5' end. And this prevents RNA2 from forming stable RNA-DNA hybrid at the origin. So all the downstream stages involving RNase H and DNA polymerase cannot occur, which means no plasmid replication if the 3' end cannot be established at the origin. If RNA2 can be thought of the sense RNA, then RNA1 is the anti-sense RNA. Typically, this sense and antisense RNA pairing is pretty weak. So to stabilize it, COL-E1 natural plasmids carry a gene encoding for a protein called ROP. ROP protein handcuffs the two RNA together. You can think of this stabilization as a negative feedback to the plasmid replication. Essentially, the increase in plasmid count stops the replication, because more plasmid means more RNA1 and ROP. therefore a stronger repression of the replication. Which means that if you plot the copy number of the plasmid for each bacterial cell division cycle, the increase in the copy number of the plasmid only occurs immediately after the division, because in the beginning, the RNA1 and ROP concentration will be relatively low, so the repression is weak. So you could probably think of increasing the copy number of the plasmid by simply mutating the regions where RNA2 and RNA1 overlap in such a way that the secondary structures are not destroyed, but it prevents the pairing of the two RNAs. A second way to increase this copy number is to delete the ROP gene. The plasmid vectors we use today depend on these changes. The first artificial vector for cloning was built in 1977. It was derived from multiple naturally occurring and synthetic intermediate plasmids. These plasmids have a copy number of around 20. The name of this artificial vector is PBr322. P is plasmid, Br are the initials of the scientists who developed this vector. Here's what it looks like. It is 4,300 odd bases long. It has two antibiotic resistance genes, one against ampicillin and the other against tetracycline. There is an ORI type of replication origin that is taken from the coli-1 plasmid. It still has the ROP gene though, so you can imagine that its copy number is not very high. Apart from this, it has many restriction sites which can be used to insert a DNA of your interest. Take note of the fact that restriction sites are spread all throughout the plasmid. The purpose of the vector is simple. It is meant for propagating and copying a desired piece of DNA. Essentially, you want to cut open a vector with a restriction site and insert your DNA of interest in that vector. PBr322 was a major advancement because it offered different choices of restriction sites. So if you have restriction enzyme, say here, A also cuts your fragment of interest internally. You could switch to some other restriction site. Anything you insert into a plasmid adds to the size of the plasmid. For plasmids, the total capacity is around 10 to 12 kb. If the vector is around 5 kb, your insert shouldn't be more than 7 kb. In summary, PBr322 is a cloning vector with many restriction sites. It is sadly not a protein expression vector. One of the issues is that it doesn't carry any screening markers, and I will explain screening markers later in the video. The copy number is still 20 because it has this negative feedback mechanism active just like the coli-1 plasmid. 4.3 kb for a vector is still quite large. So around 1982 to 1985, the PBr322 was modified quite a lot. The result was the PUC plasmid series. Versions 18 and 19 or their derivatives are the most commonly used plasmid vectors even today. The genius of PUC1819 or PUC1819 is the introduction of the polylinker or multiple cloning site. This is a short region in the plasmid that has a whole bunch of restriction sites. They usually contain a screening marker and a lot of restriction sites. Compared to PBr322, they are tiny, and the copy number is 30 times. Here's the map of PUC19. It has the origin from PBr322 and the ampicillin resistance gene. The ROP gene, however, is missing. That explains the high copy number. The highlight in this plasmid is this LAC-Z alpha. This is a screening marker. In this LAC-Z alpha, there is a multiple cloning site containing 15 plus different restriction sites. So from PBr322 to PUC plasmid, the ROP gets deleted. The RNA1 and RNA2 pairing region is mutated. This helps cut down the plasmid size and increase the copy number. There's the MCS novelty as well. PUC is still a cloning vector. From PBr322, many derivatives exist that can be constructed to drive protein expression. I will discuss them when we get to the video about cloning genes and protein expression. From PUC19, a new family of vectors was developed. This includes BlueScripts, GEMs used for TA cloning, EMBL vectors, and many more. BlueScripts are most commonly used out of these. They have an F1 bacteriophage origin of replication in addition to PUC19 origin. They look and work like plasmids, but technically they're called phagemids because they can do something that plasmids cannot. They're a mix of phage and plasmids. They have the extra ability to propagate plasmids into a virus. But apart from that, they do everything a plasmid does. Phagemids, especially BlueScript, can fit a lot more DNA while still retaining a fairly high copy number. To give you an idea about phagemids, I will take pBlueScript as an example. Remember the M13 genome from the rolling circle amplification? where it has a double-stranded origin that gets nicked by the GP2 protein in the RF2 stage. If not, you can watch the RCA video that I have, links down in the description. Well, there's a phage called F1 which has the exact same feature. This double-strand origin is not just the origin of replication, but it also serves as the packaging signal for the phage DNA to get packaged into a virus particle. So BlueScript has pretty much all the PUC19 parts, another word would be the backbone of PUC19, plus the F1 phage origin of replication. So it is a plasmid, but better. Here's a pretty map of BlueScript too. And you can notice the F1 origin is engineered within LAC-Z alpha. Essentially, everything else pretty much looks similar to PUC19. There are some differences, but let's not go into distracting details. Let's understand the superpower of a phagemid. Imagine this happy bacteria is carrying your BlueScript plasmid. You can infect this bacteria with the F1 helper phages. These will be special phages where their own origin is slightly mutated, so they cannot efficiently package their own DNA. All other proteins are fairly normal and will help in the virus production later. Once these helper phages inject their DNA into the bacteria, the DNA from this helper phage will make viral proteins. Out of these proteins is GP2. GP2 will cut the double-stranded BlueScript plasmid and make the plus strand out of it. This converts BlueScript into single-stranded DNA. The F1 origin in this state is the signal for packaging into the virus particle. Since helper phages have a mutated origin, its own DNA is not packaged as efficiently, and BlueScript wins the competition. The helper phage therefore serves only to provide packaging proteins. If you need more details on GP2 and the viral DNA replication, watch the rolling circle amplification video, which is linked down in the description. Anyways, this infection converts your BlueScript into a single-stranded DNA. Originally, BlueScripts were constructed to make strand-specific DNA sequencing libraries, and were also used in gene mapping, where direction of the strand is very important. Note that the new virus particle, which has the BlueScript DNA, does not carry any proteins to make a virus. So the new virus particles that you get from this can infect a bacteria, but cannot replicate the DNA by itself. The bread and butter of plasmid application is in selectable and screening markers. Selection or selectable markers are genes in a plasmid that permit the growth of a cell under a very specific condition. Antibiotic resistance is a classic example, and we will discuss that. Other examples include mutation suppression, auxotrophy reversal, or even synthetic lethality. Synthetic lethality is the second most commonly used phenotype. In an experiment, you're trying to insert your plasmid into a large population of bacteria. In the most simple case, two scenarios are possible. Your plasmid either ends up in a bacteria or it doesn't. And let's say your plasmid carries the antibiotic resistance against ampicillin. If you try to grow all these bacteria in the presence of ampicillin, all the bacteria that don't get the plasmid will die, and only the ones receiving it survive. In essence, you have selected for the bacteria which carry the plasmid containing resistance against the antibiotic. In this case, we're saying ampicillin, but you can have tetracycline, kinamycin, and many others depending on the plasmid. So, selection is a way to positively select your bacteria of interest. What about synthetic lethality? The two most common markers are SACB and CCDB. For simplicity, I'll focus on CCDB. In this case, the plasmid you're trying to use has CCDB gene in addition to an antibiotic gene. CCDB is a toxin for the bacteria. So, your approach with using CCDB relies on inserting your fragment into CCDB or replacing the CCDB altogether. By doing this, you will make the CCDB non-functional, which means that toxin cannot be produced. The experimental setup is the same. Because of the antibiotic, all non-plasmid containing cells will of course die. But apart from that, you have two scenarios. One where the bacteria receives the active CCDB plasmid, others where the bacteria receives the disrupted CCDB plasmid. Since active CCDB is toxic, this type of scenario means death. So you will actively select for disrupted CCDB plasmids. For your notes, CCDB is a poison for DNA gyrase, which is a class of topoisomerase. In DNA replication forks and transcription, the helix in the front undergoes positive supercoiling. DNA gyrase relieves the supercoiling. If gyrase is locked because of the toxins like CCDB, the bacteria will die. Now here's a question for you. Since CCDB is active in starting plasmids, how do you even propagate this plasmid in a bacteria without killing it? Okay, the next obvious step after selection is screening. Screening for your insert. This means checking whether the plasmid has the insert that you expect, or if it is something totally unexpected. There are different ways to screen. There are markers that allow color-based screening. Restriction digests and Sanger sequencing are the most common ways to screen as well. Colony PCR is another way to do this. Colony PCR and Sanger sequencing seems pretty self-explanatory, so I will focus on the other two. Starting with restriction digest. Let's say your starting plasmid was selected for antibiotic resistance. You tried to insert your DNA of interest between restriction sites A and B. Both of these plasmid will get selected and grow. And then how do you check if the bacteria that you have contains the desired plasmid or some fragment from the original plasmid itself? Let's define a few things. Say the distance between A and B is 200 bases versus when you insert something, the distance is 1000 bases. And in this plasmid, there are restriction sites X and Y that are 550 bases outside of A and B. Since X and Y are outside of A and B, and we only expect changes inside A and B, the outside is the same in both plasmids. And assume that the total length of the original plasmid is 2000 versus 2800 for the desired plasmid. Essentially, you expect to screen for a difference of 800 bases. The most intuitive test you can think of is to digest your plasmid with enzymes cutting at X and Y. Depending on the original and desired plasmid, the outcomes will be different. For the original plasmid, X and Y digest will give you 750 bases on the inside and 1250 bases on the outside. In contrast, the desired plasmid will have a bigger inside fragment of about 1550 bases versus 1250 bases on the outside. The so-called outside is referred as the backbone of the plasmid, whereas the insert part is variable. The unchanged backbone of the plasmid is a good way to tell that you are at least screening the plasmid you started with and not some unexpected trash. This is great, but what if there was a possibility that the insert you expect is not actually present, but a different 1000 base insert somehow got in? The X and Y digest does not differentiate the not desired plasmid from the desired plasmid. Both have 1000 bases, so the outcome of X and Y will be identical. The simplest way to screen this difference is to select a site which is present in your DNA, which hopefully is not present in the original plasmid. Let's say you found Z, which is expected to be around 400 bases away from A. Now, if you do X plus Z digest, you will cut the original plasmid only with X, producing a linear plasmid of 2000 bases, versus 900 bases from X to Z in desired plasmid and the remaining 1900 as the second product. The undesired plasmid is unlikely to have the exact same patterns of cuts as your desired plasmid. This type of strategy is extremely useful if you were trying to replace the 200 bases in the original plasmid with the new 200 base DNA. In that case, you surely want to find a site Z in your DNA. Another popular way of screening is color-based. You can do this via fluorescent proteins in your plasmids, and that means your bacteria may glow in a specific color. LAC-Z alpha is probably more widely used, so I will limit the discussion to this. We saw this in PUCs and BlueScripts. I also hope that you recall LAC-Z from lac-operon, because this is it. Okay, so you expect that selected bacteria is carrying LAC-Z alpha, but now you want to tell whether or not it carries your expected insert. We noticed that LAC-Z alpha had this multiple cloning site, into which you typically try to insert your DNA of choice. So there are these two expectations in a positively selected bacteria. The bacteria used for this is special. It carries a mutation in the LAC-Z, so the bacteria is deficient and cannot make LAC-Z enzyme. The LAC-Z alpha of the plasmid, however, can help recover the bacteria from this deficiency. We call this alpha complementation. The LAC-Z alpha complements LAC-Z mutation. By the way, this method of screening is popularly known as blue-white screening. The idea is simple. You expect selected bacteria to either have a pristine LAC-Z alpha or an interrupted LAC-Z alpha. If the alpha is active, it'll make a short peptide. This short peptide helps the mutated LAC-Z to recover and become functional. If the alpha is interrupted because you inserted some DNA in it, this will fail to rescue the bacterial LAC-Z. So how do you distinguish these two possibilities? These bacteria will be fed two substrate called XGAL and IPTG. A functional LAC-Z will convert XGAL, which is a type of galactoside, into a blue color pigment. IPTG is an analog for allolactose, so it induces lac operon. This blue color pigment will be produced in bacteria containing a functional LAC-Z. This happens if the alpha peptide is produced, and this happens if the insert was not inserted in the plasmid. In the other case, the LAC-Z is non-functional because the inserted DNA broke the alpha gene, so it cannot use XGAL, and therefore these bacteria do not produce any color, and they have their own natural whitish color. This way, you can differentiate the bacteria based on colors. Here's a picture of how it looks in reality. This video is only to give you an intuitive and conceptual sense and relevant background on how plasmids are used. I will give you a much more detailed view of inserts, restriction digests, selection and screening, and how to actually use them when we get to the videos on actual molecular cloning. As always, I will see you in the next video.